Assalamu alaikum. So in this lecture, what we will do is we'll go over a new technology known as the CRISPR Cas9, and it's used in gene editing. Okay, so let's go on with this amazing technology that has changed the world and will change the world definitely. So what does CRISPR stand for, and what is Cas9? So CRISPR is the abbreviation for clustered, regularly, interspaced, short, palindromic repeats. Okay, so basically what we have here, you know what palindromic means now, right? So these are the palindromic repeats. Okay, and they are, as you can see, interspaced. So there are spaces between them, and they are regularly organized. Okay, and they are clustered in one area in bacterial DNA, in the bacterial chromosome. So this is a bacterial genetic system. The main purpose of CRISPR is to fight bacteriophages. Okay, now Cas9 is a nuclease, so it is an enzyme that degrades nucleic acids, and it is RNA guided. So it binds to, it associates with an RNA molecule, and this RNA molecule takes it to a certain place in the DNA. So it is a ribonucleoprotein in that sense. Now, um, the RNA molecule is actually transcribed from the CRISPR system. These are the colored regions right here. So the black are the palindromic repeats, and the colored regions are portions of the bacteriophage DNA that gets inserted into the bacterial chromosome. Okay, and they are transcribed into an RNA. The RNA associates with Cas9, the nuclease, and the RNA guides the Cas9 to certain regions in the bacteriophage DNA. Now, this is what CRISPR-Cas9 does in bacterial cells. So you can read this, but I'll describe it right here. So what happens is that you have bacteriophages, viruses that infect bacteria. They infect bacterial cells, okay? So they sit on the plasma membrane of bacterial cells, and they insert their DNA, their genetic system, into bacterial cells. Now, and they take over the whole system, okay? And then they multiply inside cells, and cells eventually rupture, releasing all of the lot of bacteriophages that would travel and infect neighboring cells and so on. So that's how they multiply. So bacterial cells need to protect themselves. So what they do is that they're able to degrade or to take part of the bacteriophage DNA, and they insert this DNA into the CRISPR system inside their own chromosome. Okay, so here you have the palindromic repeats, and here you have part of the bacteriophage DNA. Now, this here in blue is actually a bacteriophage that infected bacterial cells previously, and again, bacterial cells took part of its DNA, the phage DNA, and inserted it into its own chromosome. Same thing with the green DNA right here, and so on. So what happens is that you have another bacteriophage, the same one as this one right here. This bacteriophage infects the bacterial cell. Okay, so it's like an immune system. This is the memory of the immune system of bacterial cells. But this bacteriophage is not successful in infecting cells. The reason is that the CRISPR genetic system is transcribed into RNA, and part of this RNA is taken, okay, so it's processed, and part of it is taken and it associates with the Cas9 enzyme. Okay, and then it guides the Cas9 to the bacteriophage DNA. So here the RNA is complementary to the bacteriophage DNA. So it associates with it, and Cas9 degrades and cleaves the bacteriophage DNA so that it cannot infect the bacterial cell. So this is again the immune system of bacterial cells. Now, that's CRISPR Cas9. So let's go back to DNA repair mechanisms in human cells. There are different mechanisms in human cells, so this is something different, okay? And then I'll connect things together. So in human cells, DNA can be repaired if it gets broken. So if you have double-strand cleavage or break in human DNA, you have activation of two different repair systems. The first one is known as homologous recombination. Okay, so what happens is that bacterial cells, in human cells, the repair mechanism, you have certain proteins that use the complementary chromosome, right? So in every cell is diploid, right? You have chromosome from Baba, another chromosome from Mama, and let's say that that chromosome is cleaved, right? And the system utilizes or takes advantage of the complementary of the homologous chromosome. It uses it to fill in the gaps. Okay, so you have now the chromosome that is broken is repaired. Now the other mechanism, repair mechanism, is known as non-homologous end joining. Okay, so in this system, you don't have the use of the homologous chromosome. Rather, what happens is that you have proteins that glue the broken DNA together. Great. So these are the two systems. So again, what you have in here is that you have the guide RNA. It contains region that is complementary to the DNA. This guide RNA binds to Cas9, and Cas9 can go to a certain region in the DNA, and it can cleave it. Okay, so what we can do is that we can transfect cells with a plasmid vector that contains the guide RNA, and it also contains the Cas9 gene, the bacterial gene. So we can introduce both of these into human cells. So the RNA would guide the Cas9 protein to certain region in the human chromosome, and Cas9 would cleave it. Okay, so that's the idea, is quite genius. And the two scientists who came up with this brilliant idea got the Nobel Prize in 2020. In fact, I should say that the CRISPR-Cas9 system was discovered by a Portuguese scientist in bacteria, except that his thinking did not go beyond what the CRISPR-Cas9 does. These two scientists utilized the CRISPR-Cas9 to do something that is quite genius and revolutionary. But here's what they did. So basically the idea is that they would introduce breaks into the DNA, and you will have by Cas9, okay, and you will have activation of both or one of the DNA repair systems. Now, if you have activation of the non-homologous end joining system, scientists have noticed that when this system is activated and you have gluing of the DNA, you will have introduction of indels indels stands for insertion deletion mutations. So you will have, if you have a gene right here and you have insertion or deletion, you're knocking out the gene. You are disrupting the gene. The gene becomes dysfunctional. Okay? Now, if you have activation of the homology repair system, uh, since we have a homologous chromosome in our cells, you would have replacement of the deleted gene, or the damaged DNA. It would be replaced by fragments from the homologous chromosome. Now, imagine if you have a mutated DNA in here, and this DNA is good. There's no mutation. So what you're doing is you're correcting the gene. You are repairing it by replacing the mutated DNA with a good DNA. Well, it can be the opposite. You can have a good gene right here, and what you do is that you are replacing it by a mutated DNA or gene. So you can as well fool the system, and you can insert a new DNA inside the chromosome as well. Inside this DNA right here, you can insert a whole gene. So the potential is really amazing with what you can do with the CRISPR Cas9 system. So this is how it's done. These are the uses of the CRISPR Cas9. So basically, you can introduce Cas9 and the guide RNA into human cells. The guide RNA takes Cas9 to a certain region in the DNA, and Cas9 cleaves the human DNA. You have activation of the non-homologous end joining, and you can introduce indels, insertions, or deletion of the DNA. And that eventually leads to frame shift mutation if we're talking about a gene that is mutated. Now, if you have activation of the homology directed or the homology repair system, you can introduce a new piece of DNA, you can correct a mutation, or you can introduce a mutated piece of DNA. Okay? And this is really the act of manipulating DNA. This is how this is changing the fingerprint of DNA. I love these images. You can insert a piece of DNA of your choice. You can do genetic engineering. You can play around with DNA. You can cut it and modify it. You can repair it. And they're thinking actually of introducing new pieces of DNA into our cells. So this is the new drug in mind. Now, I'll end with these stories, two stories. This is, of course, not for you to learn in the exam, but you can go back and read about these news. So here you have a scientist in 2016 who came up with an idea of using the CRISPR-Cas9 to mutate genes in the fertilized egg, in the zygote, hoping to learn how the zygote becomes a baby, how it differentiates. And of course, it made a huge stir. It was so controversial that there was a lot of talk about whether her research should be funded or not. Eventually, it was funded in the UK because she said that she wouldn't go beyond 2 weeks post-fertilization. So it's amazing what this can do. The other piece of news is even worse. Now, in 2019, a Chinese scientist used the CRISPR-Cas9 technology to change DNA of 2 baby girls. Well, before birth, of course. So he manipulated a gene inside the zygote, and the twins were born with the edited gene. Now, the gene that he changed was CCR5, and this gene would protect babies or protect human cells from the HIV. So HIV, the virus, cannot get into the mutated uh, cells. Now, the thing with CCR5 is that it's also linked to intelligence. So if it's not there, there's an assumption that it increases intelligence. Now, does it do that or not? I have no idea. Um, Now, the thing with the scientist is eventually there was a lot of controversy all over the world, and he got jailed for 3 years, apparently, for doing something that is, just it's a red line that he crossed. You cannot do that. Ethically, this should not be done. But, you know, the potential is there, of course, and this is the future.